there are many ways to get different alleles. We saw all these types of mutations earlier and how genetic linkage is affected by things such as inversions and duplications. Now, let's look at heterozygosity from a population perspective and mutations from an organismal perspective. For this, I'd like you to be able to measure heterozygosity. List the 4 types of mutations, keep in the context of the fitness of the organism. Remember, fitness is the number of offspring produced relative to others in the same species. And explain how natural selection removes deleterious mutations. Perhaps you have heard the question, if most mutations are bad, why does mutation lead to evolution? This is the lecture where you learn that. So, heterozygosity. Heterozygosity is overall how many times you're going to find two PQ in a population. So how often do we see 2 PQ for an allele? Why does it matter? Well, there can be something like a heterozygote advantage, where the organism that has both alleles is going to have some sort of advantage in fitness over others that only have one allele, albeit 2 copies. Heterozygosity is also a measurement of inbreeding. So a population that has been highly inbred has a lot of homozygotes and not a lot of heterozygotes. Heterozygosity is also a measure of genetic diversity. So how many different alleles exist and how many different alleles are going to be heterozygous in a population is a measurement of how diverse the population is. How do we measure this? Well, there are allozymes. So allozymes are going to be on one of these gels. Remember, running a gel, you load perhaps a protein mix into here, and here we might have a homozygous dominant individual. Here we might have a homozygous recessive individual, and an allozyme is going to have that, 2 separate bands. Allozymes are separate enzymes, so other enzymes, and what you're going to see is 2 bands for the heterozygote as opposed to 1 band only for each homozygote. There's also the possibility of doing sequencing and gene sequencing individuals to see if they have a certain allele or not, or if they have 2 copies. If in a genetic sequence you have 2 different nucleotides at a certain point, that is a point mutation. Remember, we did point mutations in the previous lecture. And if you have that point mutation, you have those A or G. It's either A or G. It could be that it's both A and G in that individual because it's heterozygous. So that's a way of sequencing and gene sequencing to find heterozygosity. This is kind of how that's measured. And we also see that's measuring. We can measure that as mean heterozygosity. So what fraction of the population is going to be actually heterozygous for a certain allele, or for overall a whole bunch of alleles. So how often do we see heterozygosity in a population? What is the percentage of loci? So if you have, for each gene, what's the percentage of those that even have different alleles? So for this gene, there might only be 1 allele. Okay, it's not heterozygous, but for another gene, there might be 2 different alleles. For a third gene, there might be 3 separate alleles, like the ABO blood types. So how many different loci are polymorphic, i.e. how many different loci have different alleles? These are ways of measuring it. The effects of this are pretty clear and simple. It's variation. And if that variation exists in phenotype, it is something on which natural selection can act. So we're looking for heterozygosity as a way to measure variability that is inherently heritable because it's genetics. So this is kind of covering some of the early bases of evolution and determining how well can this population evolve. If you've looked into invasive species, you'll know that there's something called a lag phase. If you introduce just one type of plant, let's say it's a kudzu vine, and it's going to grow and self-propagate, but it's the only one on that continent, it isn't, if it's unable to reproduce with itself, then it has a very low genetic diversity. And that's actually going to affect its ability to adapt to changing environments. But if you have a large amount of genetic diversity in an invasive population, then that genetic diversity can yield itself to allow the population to evolve. So we're measuring this as a potential for evolution vis-a-vis the amount of variability. All right, mutations. Mutations are going to have fitness effects. or they're going to have no fitness effects. Either way, they're going to have something. Either something or nothing. Sorry, that's not very specific there, but let's talk about exactly what I mean here. There are different types of mutations you're going to get, and the question is, if most mutations are bad for the protein, how can evolution proceed if so many mutations are deleterious or lethal? Basically, there are so many ways to screw up a protein. How could it ever become better? So what we see is how bad are mutations really? So we do something called mutation accumulation. It's when you put the organism under perfect condition and allow mutations to accumulate in the absence of natural selection and see if the fitness is going to increase as the mutations increase. Perhaps you can just mutate your way to a better organism if there's no natural selection. Kind of an odd way of doing things. But that's one way of determining also what the rate of mutation would be in the lack of natural selection. So as we see here is a whole bunch of different organisms and how frequently they have mutations. You can see down at the bottom, human and mice have somewhere between 10 to the negative 4 and 10 to the negative 3 substitutions per base per generation. So other organisms such as HIV have very high mutation rates, and some organisms like Halofirax volcani have very low mutation rates. So there's different mutation rates for different organisms, which tells you how fast they can evolve. And how we determine that by how many mutations they'll accumulate given no natural selection. But there is natural selection. And there are types of mutations in the presence of natural selections. 4 types. 4, not few. We lump these together in 4 different types of mutations. The first one should be pretty self-evident. It is lethal. This is a mutation where the cell doesn't function. Boom, the cell is dead. Or the organism cannot function. Boom, the organism is dead. And a lot of mutations are lethal. A base pair substitution that results in a premature stop codon for a gene that is absolutely essential for survival, that kills the organism. Game over. Hope you saved. Lethal mutations are gone out of the population pretty quickly. Then there are deleterious mutations. What you see on this graph is mutation accumulation lines where they just let the mutations accumulate in the absence of natural selection. So lethal mutations can't accumulate because they kill the organism. So we're already saying those aren't going to be shown in this. What we see is fruit number here, a measure of fitness for Arabidopsis thaliana, Thalcrest. For most Arabidopsis, they're going to have between 16 and 22 fruits. And that's what you see in these pre-mutation lines. That's the very few lines kind of at the very bottom. It's an odd graph. But those lines had between 16 and 22 fruit per organism. Now you let these mutations accumulate, and you get some of these mutants are only having about 10 or 9 fruits per organism. Those are ones that have accumulated some very deleterious mutations. They're not dead, that'd be lethal, but they're not doing very well, so that would be deleterious. But we see there are a lot of neutral mutations. So these neutral mutations we see as it's almost a normal distribution. If that 16 was higher, it would pretty much be a normal distribution around the pre-mutation lines. So most mutations are normal. If we're seeing a bimodal distribution, that would suggest that most mutations are deleterious or beneficial. But realistically, most mutations are neutral. No change. And that's going to just be as is. It's some sort of mutation that occurs outside of a gene. Or it's a mutation that doesn't change an amino acid. Or it's a mutation that changes one amino acid to another, but it doesn't really matter because you're changing, valine to isoleucine. Okay, they're both very similar. You're not changing valine to arginine. Ha ha ha. You should know your amino acids. And last, there's beneficial. Now this graph might fool you. You see there are several lines where you have more than 22 seeds. So these are some plants that are able to produce many more fruits. Sorry, not seeds, fruits. These are plants that are able to produce more fruits. So you're thinking, oh good, those are beneficial. And it looks like beneficial mutations are pretty common too. Well, anything less than 16 is a deleterious, and you can see a lot of deleterious mutations. Anything that's greater than that 20 or 22 is a beneficial mutation in the absence of natural selection. So it could be that this is a mutation that allowed the Arabidopsis to put more energy towards seed production, but in the absence of being able to defend itself against pathogens. Oops. That's in the presence of natural selection, that organism will be dead. In the absence of it, that organism will be doing fine. So remember, not all beneficial mutations are universally beneficial, just like not all deleterious mutations are universally deleterious. All right, so a benefit of multicellularity I kind of glossed over here is the organism can use cells without lethal mutations. So let's say that the Arabidopsis line here that's going to be exposed to a mutagen, one of the cells gets a lethal mutation. That cell's dead. Okay, you lost a cell. Moving on, oh no! But anyway, it doesn't matter if you lose a single cell if you're a multicellular organism. A multicellular organism allows that organism to essentially get a lot of cells that get the lethal mutations and never worry about the total consequences of that, and only really deal with the deleterious, neutral, or beneficial mutations. So that's in the absence of natural selection. You can have deleterious, neutral, or beneficial mutations continue through a line, provided nothing's stopping that line, like returning natural selection. So here is the ratchet. You have this group here of Heterozygosity elegans, these nematodes, and what you have is you allow it to just take on more mutations. During mutation accumulation, you will have a lot of them that are going to be getting a lower survival to maturity. Your control group, as you see, is just, well, it's control. It's around 90% surviving to 97% survive to maturity. Okay, cool. During mutation accumulation, where you do not have natural selection, you actually get more deleterious mutations occurring. And this is what you would say if someone were to say to you, how can evolution by natural selection actually result in better organisms if most mutations are deleterious? Look, in the absence of natural selection, most mutations are going to make lower survivorship in this Heterozygosity elegans. Yes, but in the presence of natural selection, you see it go right back up. Right back in a very few generations, 60 generations, you see that those lines recover back to normal. What happens here is that death is going to cull all lethal mutations. You don't see them. That's not part of our story here. Lethal mutations are gone. And there are 4 types of mutations now. There are 3. There's deleterious, neutral, or beneficial. Most mutations are going to be deleterious. Okay, but natural selection gets rid of those. So in a real population of organisms existing out in the real world, deleterious mutations very quickly disappear. In inbred populations, in endangered species, the deleterious mutations might actually persist just due to a lack of genetic variability. But in the real world, big populations, Hardy-Weinberg, whatever, with mutations, then most deleterious mutations are going to be gone in a generation or 2. Only neutral and beneficial mutations can accumulate. So that means neutral mutations will accumulate at a large rate. We're going to come back to this with something called neutral theory, and it is going to be a brain buster, but a really good one. But this kind of ties in. If you think this part through, then neutral theory makes a lot more sense. Only neutral and beneficial mutations can accumulate. Most mutations are going to be neutral. So a very few will be beneficial. Now I want to picture it this way. Let's say that everyone buys lotto tickets, and there are winners chosen and there are losers chosen. And when I say losers chosen, what I mean is if you have a certain number, then you can no longer buy lotto tickets. Neutral mutation means you can buy 1 lotto ticket per day, and you can buy 1 lotto ticket the next day. A deleterious mutation means you can buy 1 lotto ticket today, but no lotto tickets tomorrow. Lethal mutation means you buy a lotto ticket and then it gets stolen from you and ripped to shreds. I don't know. It doesn't play. They're not playing. Deleterious plays once, doesn't play again. Neutral can play once per day. But the beneficial mutations are those people who win the lottery and thus can buy more lotto tickets. Perhaps they only win 5. Okay, a person who buys 5 lotto tickets and buys them on the same number is going to win 5 for each one of those 5. That's what a beneficial mutation looks like. A beneficial mutation is when the organism is going to have a higher fitness in the next generation, and all that have that mutation will have a higher fitness. So you're imagining your population of people, some of whom are buying lotto tickets and that are out of the game, the deleterious ones. Some of whom are buying 1 lotto ticket per day, every day, and the same amount. That's your neutral mutations. And some of whom win a small amount of money, which lets them buy more lotto tickets, but all of those also win a lot of money. And you see very quickly that person who bought that winning ticket, that got that beneficial mutation, whatever numbers they're playing, they'll play the same numbers the next day and win the same amount the next day. They're very quickly going to be buying many more lotto tickets. And if there's a limited pool, say only 1,000 could be bought, it won't take long before those beneficial tickets are going to be taking all of them. And that's what beneficial mutations will do. They make the organism able to have higher fitness, and that beneficial mutation sweeps through the population until it dominates the whole population. Beneficial mutations go to fixation quickly. So that's how we have it. How can evolution by natural selection proceed if most alleles are lethal or deleterious? Because some are beneficial. The lethal and deleterious ones go away, but the beneficial ones move up and then move up. And you can accumulate these over generations because every time a new one comes in, it ratchets up through the population quickly. We'll see that again with something called selective sweeps, with something called selection gradients, and we'll see neutral mutations do accumulate as well through chance alone in what we call neutral theory. So you will want to know these 4 types of mutations because we're going to be going quite a good distance with them. Enjoy.